Karibu kwenye channel ya Dr. Mlelwa Lectures. Tunafundisha masomo ya sayansi, physics, chemistry, mathematics pamoja na biology kwa syllabus ya Tanzania na kwa O-level pamoja na advanced level. Dr. Mlelwa Lectures, for your better NECTA EXAMS performance. Subscribe, share and like this video. Welcome back, friends. In the last session we looked about the functions of bile in the body, and in this session I want to continue with the function of the pancreatic juice during digestion of food in the small intestine. So I hope you remember we discussed about the function of bile salts, that is emulsification, breaking up fats by the process called emulsification, and that is physical digestion, as we said yesterday. Emulsification in the breakdown of fat globules in the duodenum into thin droplets which provide large surface area in which enzymes of the pancreatic lipase can act to digest the fats into fatty acid and glycol. So emulsification is the physical breakdown of fat globules into small droplets by the bile salts so as to increase the surface area for pancreatic lipase to act. This is another definition for emulsification. But one thing you should note here, that emulsification is the physical digestion and not the chemical digestion, as most of people think that emulsification is the physical digestion. And we will look about the importance of the bile in the digestion. Now let's move to the pancreatic juice. As we know that the pancreas perform two functions, that is, it secretes exocrine hormones or secretions, and it secretes endocrine hormones. Exocrine secretions, they are pancreatic juice, collectively called pancreatic juice, and the endocrine secretions, it is insulin hormone. So it is insulin and the glucagon hormone. All of those, they are involved in the control of the blood sugar level. They are involved in the control of the blood sugar level. Now here we are discussing about the exocrine secretion, that is pancreatic juice. Now within the pancreas there are a group of cells which will produce a variety of digestive enzymes that are poured into the duodenum via the pancreatic duct. The pancreatic juice, in addition to water it contains, first is mineral salts, which are sodium hydrogen carbonate. Pancreatic juice contains sodium hydrogen carbonate. This is important for neutralizing the acidic chain from the stomach, and also create a more neutral pH for the enzymes of the small intestine to work. So most of the enzymes of the duodenum, they work at the neutral pH of around 7, and we know that the acidic chain from the stomach had the acidic pH around 1 or 2 pH. So that's why we need more bicarbonate ions, and one among the way of providing these bicarbonate ions is through the secretions of pancreatic juice, which contain the mineral salts. We shall see later in the nervous control that there are some of the stimulation which stimulate. There are some of the hormones and nerve impulse from the nerve endings at the wall of the duodenum, which send the impulse to the brain, and then the cells, they secrete more bicarbonate because of the entrance of the acidic chain. So we shall see later, just stay with me. And then we are seeing that we have proteases. These are enzymes involved in the digestion of proteins. Proteases. In the pancreatic juice we have almost 3 types of proteases. First is the trypsinogen. Trypsinogen is inactive enzymes, like how pepsinogen and the prorenin, they appear in the stomach. So trypsinogen, which when activated by the enterokinase from the intestinal wall, form endopeptidase called trypsin. So trypsinogen is activated by the enterokinase. Enterokinase is also an enzyme, and enterokinase is produced by the cells of the intestinal wall. So trypsinogen from the pancreas, it is inactivated until when it reaches the lumen of the small intestine in the duodenum. There it is where the enterokinase from the intestinal wall tend to activate the trypsinogen into trypsin, and trypsin function, which hydrolyzes protein into peptides. Trypsin also activates another protein in the secretion called chymotrypsin into chymotrypsinogen into chymotrypsin. So trypsin is activated by enterokinase, but trypsin itself, it activates another protein called chymotrypsinogen. So you see, ni kama cascade of events, kwamba mmoja na activate mwenzake. Ni kama ilivyo kwenye blood clotting mechanism. Blood clotting mechanism, unakuta ni mmoja ana activate mwenzake until at the end of the day yukuta amefika point ambayo blood clotting inatokea. Lakini kama unavyoona hapa, trypsinogen anakuwa activated by enterokinase, then trypsin, which is activated trypsinogen, inaenda ku activate chymotrypsinogen into chymotrypsin. Chymotrypsin pia yenyewe ni ina convert protein to peptides. Pia there are also another exopeptide called carboxypeptidase, which convert peptides into smaller peptides and some amino acids. So as we studied in biochemistry, carboxypeptidase, they are peptides which break the peptide chain, which break the polypeptide chain at the carboxyl end. So that's why I taught biochemistry first before nutrition. So if you don't understand about the carboxypeptidases, just go to review the session of the classification of enzymes. Also go to review the session of the formation of dipeptides when he was discussing about the structure of amino acids, and then you will understand what is the meaning of the carboxyl end and the meaning of the amino end of the polypeptide chain, and then it will be easier for you to understand about endopeptides and exopeptidase. Endopeptidase and exopeptidase. Endopeptidase, they always break the bond between the amino acids within the polypeptide chain, while exopeptidase, they break the terminal bonds at the carboxyl end and at the amino end. So the endopeptidases, exopeptidases, breaking the bonds at the amino end, they are called aminopeptidases, while the exopeptidases breaking the bond at the carboxyl end, they are named as the carboxylpeptidases. And now let's move to the pancreatic amylase. A pancreatic amylase completed the release of starch to maltose, which began in the mouth. So this function as amylase in the mouth, salivary amylase, how it does, it hydrolyzes starch to maltose. And then we have lipase. Lipase, or sometimes it's called pancreatic lipase. This break down fat into fatty acids and monoglycerides, yani glyceride from one fatty acid by hydrolysis. So it breaks down fats or lipids into fat acids and monoglycerides. So we shall see later how are these absorbed, because according to our biochemist concept, you could expect here to end up with the fatty acid and the glycerol, but wondering enough here that we are ending up with the fatty acid and the monoglycerides. We shall see later how are these monoglycerides and fatty acids they absorbed into the body, and how are they incorporated to be transported into different parts of the body, because the absorption and transportation of lipids to different parts of the body is quite different from the absorption of other kinds of food. And then we have nucleus, which converts nucleic acid into HC constituent nucleotides. So I'm ending up here. In the next session we shall be looking about the intestinal juice components and how it functions. Thank you. Asante kwa kuangalia channel ya Dr. Mlelwa. Ninaamini umeelewa kipindi, na kama hujaelewa chochote waweza kucomment hapo chini. Click link ya kwenye description kujoin Telegram group kwa ajili ya discussion. Usisahau kusubscribe, kushare video kwa marafiki pamoja na kulike video hii.